And so here's the idea behind the label transfer experiment. Okay, so the hypothesis is that the activation surface on CRP, this could be AR1, then makes a stimulatory contact with RNA polymerase. So what the label transfer experiment attempts to show is that a particular region of CRP is in direct contact with RNA polymerase. And so the way you do that is you label that region in CRP that you're studying, in this case AR1. You put some kind of a transferable label on CRP, and then you induce the transfer of the label onto RNA polymerase. Okay, and so the labels are designed in such a way that this transfer only occurs if these two protein surfaces are in direct contact with one another. Okay, and so if you see label transfer, then you've basically proven that this contact occurs. Okay, so here's this structure of CRP again. In order to do this experiment, the first thing that was done was that CRP was mutagenized. So in CRP, there are, I think maybe, in wild-type CRP, there are something like 3 cysteine residues. The first thing they had to do was to get rid of all of those naturally occurring cysteine residues for reasons that will be clear in a minute. And so they converted each of those naturally occurring cysteine residues to a serine. Basically, when you convert a cysteine to a serine, you're just replacing a sulfur atom with an oxygen atom. So it's a fairly conservative mutation, and the hope is that it would not disrupt the function of the protein, and they confirmed that those mutations did not disrupt the function of the protein. Okay. And then what they did was they introduced a new cysteine residue right into AR1. Okay. So they made another mutation, this time in AR1, that converted a residue that was not initially a cysteine into a cysteine. Okay. And they once again then confirmed that that did not interfere with CRP function. Okay, so why did they want a form of CRP that only contained a single cysteine residue in AR1? It was because their labeling used cysteine-specific chemistry. So here is then the mutant CRP, which now contains just one cysteine, and so a cysteine side chain contains this SH group. So it contains just one SH group in the entire protein. Okay. And then this is the reagent that they used to label CRP. Okay, so it has this complicated name. We'll just call it IAC. Okay. And so IAC then contains this thiopyridine component. It contains cysteamine. Actually, from the sulfur to the nitrogen, that's a residue of something called cysteamine. Okay, and then the cysteamine connects the thiopyridine now to this modified salicylate residue. Okay. And the important modifications to the salicylate residue is that we have this azido group here, this N3 group here. And another important modification is that we have an iodine here, and the iodine that's installed here is I-125. It's a radioactive isotope of iodine. So this asterisk here just means we have a radioactive isotope of iodine. Okay. And so now they can just mix these two things together, and under the correct reaction conditions, then disulphide exchange will occur. All right, so in disulfide exchange, then you just have a disulfide bond. Ooh, sorry. Okay. And you react that with
the
the
videographer must be asleep at the switch because he failed to capture what I was writing on the board. Here's what I mean by disulfide exchange. We have a thiol reagent that attacks a disulfide bond, and we end up replacing one disulfide linkage with another disulfide linkage. Okay, and so in this example here, then the SH group on the cysteine side chain of CRP attacks this sulfur here. The leaving group then is thiopyridine, and we end up then radiolabeling CRP.